been doing at the University of Venda and with other collaborators. Prof, you may come and share with us your presentation. Thank you.
Morning. Yes, after such an introduction, let me thank the LOC for inviting me to give this talk and SAAB in general. This has been my family since, and you know, I love this atmosphere always of SAAB, so thank you very much. And of course, today I'm going to talk about one of the plant species that is so close to me in terms of research, that I've been working on it for some time, also with my students, particularly one former PhD student. I don't know if he's here, Dr. Tiawon. If you are around, maybe you can just raise a hand, but he's already now with Northwest University. So Dr. Tiawon also participated in the research around this particular species, as you will see as I proceed with my talk. So I'm going to approach this talk by looking at the plant ecology in terms of utilization, because that is how most of this research emanates. You'll see people using a particular species, and then you raise some questions. Why are they using this particular species in this fashion? Before you can do your ecological studies, then you go in and then try to find the reason behind such an impact on a particular population. So this is the population. For this purpose, I just term it population ecology through a prism of sustainable practice, a case of Brachyregia zangobarica. So, as a way of background, it's important to indicate that population ecology is based on historical utilization. I like this approach in the sense that it really justifies why it is important to look at utilization before we can embark into the population ecology studies. It's seen as an organized way in which scientists report what they are observing or their findings after doing population ecology. So it's just a way of packaging and report the information to the communities of interest. And then, of course, usually emphasis will be on individuals, looking at the group of those individuals, maybe the population of a particular species, and then try to look at how it is surviving, and also looking at the reproduction thereof and the relationship with other plant species. So at the end of the day, this kind of data should be able to assist us in forming a basis for landscape and ecosystem studies. So indeed, populations within biodiversity play an important role, and then it makes the biodiversity tick. So in other words, it is important to maintain biodiversity in ecosystem so that we can continue to get the provision that comes out of the ecosystem, be it the natural resources as well as the services thereof. So it is important that we monitor the way in which we are using this biodiversity. You know, humans have footprints which sometimes can be negative as we leave them behind. So it is important so that indeed we can be able to focus on those species that we really need to make sure that indeed they form part of our conservation strategies, so that again we continue to enjoy the benefits. So there are several factors that normally play a role in terms of determining whether the species survive or whether the species is driven into extinction. So these are just some of the factors that we really need to look at. Exploitation and sustainable use of those resources, expansion of agricultural activities, uncontrolled fires, it's quite a number of them, settlement, you know. These are some of the things that really, as much as they are necessary for us to survive, we should also know that it is important that we don't really suppress some of these important species and then render them rare or endangered. And of course, this thing is usually driven by our harvesting pattern as we continue to use those natural resources. So we really need to make sure that our harvesting pattern do not really impact negatively on the structure of population of particular species. So, and actually if you look at it, the issue of harvesting depend on a number of factors. These are just some of them. What are the paths that are being harvested? I mean, over the days when we were attending this conference, we heard how people are using barks and roots. You know, they play a very important role in terms of determining the trajectory of a species, the paths that you are going to harvest, the life form of the species. What kind of a species is it? Is it a tree or a shrub or a herb? You know, sometimes you might find that, I mean, uprooting herbs might not be a big thing, but if you uproot trees, it's a problem. If I can just share with you, the other time I visited a population of Secrodaca along a pedunculata, and then when I visited it, I was with a traditional healer, and then he wanted to collect some roots, because anyway, he wanted to make sure that he has got something to take home with for his practice. You know, he spent a number of time trying to dig around those individuals which were there, and he couldn't find any lateral root. And then 2 years down the line, I went back to the same population, and there was nothing. So you can imagine uprooting a tree as compared to uprooting a herb. So life history of species is very much important. Season of harvesting is also very much important, particularly, especially for medicinal purposes, because people prefer to harvest, these traditional healers, they prefer to harvest during winter because of their reasons. And of course, during winter, we know already that the plants will be already experiencing harsh condition, trying to survive by all means, you know. So it's like, again, adding more pressure on the species, as well as also the intensity of harvesting, wherein people develop a tendency of ringbarking the trees. So those are some of the factors that can indeed play a major role in terms of determining how the population goes. So it is therefore important that we understand all these things so that indeed we can be able to come up with a management system that can be, in a way, integrative. So particularly when we look at the population structure, of course, there are a number of parameters that you can look at in order to understand the population structure. Usually from you in terms of the structure of the population. So, and actually when you look at that kind of distribution of size classes, then the interest then for a population to be viable, then we'll be looking at the young individuals within that population, whether we have got enough individuals that can take the population forward. So that is very much important. I know in mostly people think that a normal sigmoid graph it's one which is very much normal, and they tend to think that it means it is healthy if you are looking at a population. But nevertheless, with plant population, the graph should be some kind of inverse J shape, because they will be having a lot of young individuals, and then it should dip down as you approach the big size classes. And when you look at all these things, in other words, if, like I'm saying, if you have got scarcity in terms of seedling and juveniles, then you have got a problem in terms of the movement of your population. This particular slide of goals of conservation is very much important. I think that should always be our departure point when we try to come up with a conservation intervention. You know, it talks about the conservation of natural resources through, I mean, this was a treaty which was signed in 1982 somewhere in Brazil, in Rio de Janeiro, wherein in that treaty, what is interesting is that they came up with these 3 goals to say, as we do conservation, let us also make sure that one of the goals is to make sure that we conserve the biological diversity. That's very much important. That should be the center. And then also sustainable use of its components. And I know at times as scientists, we try to shy away from this particular goal, but it is a goal nevertheless, and we have to observe it. Whenever you are a manager of a conservation institution, maybe in resources, but in a sustainable manner. You see, because of course, if you just say this thing to communities, they will forget about the sustainable aspect and focus on the use. But we have to make sure that when we use the components of biodiversity, we use it in a sustainable manner. That is being, thinking forward in terms of the future generation so that we can preserve for them to also enjoy the same benefits that we are enjoying. And of course, there are issues around fair and equitable sharing of benefits that are arising from genetic resources. That's also straightforward, that we need to share the benefits. So it can be somewhere, somehow, when you find that you got benefits of resources maybe from a particular community and you run away with the benefits. You don't want to share with the community. That will be something somewhere which need to be looked at. So I'm saying this thing and then bringing again the United Nations Sustainable Development Goals. That was in 1982, and this is quite recent, wherein they're talking about 2030. But in those goals, goal number 15, that's the one that I decided to pick. It talks about protecting, restoring, and promote sustainable use. What I like about this goal, again, it emphasizes the issue of use, which is, again, sustainable. So we need to really, as we approach our research in terms of conservation, look at these goals and then check whether indeed are we contributing in terms of achieving these goals. So by then, when you look at all this, then the big question becomes, what makes certain species more important than others? You know, what is it that makes you want to focus on particular species and maybe not giving same attention to other species? Then this brings me to the story about this Brachyandrigia zangobarica. This is, of course, a plant which belongs to the family Ochnesi, and it is commonly known as yellow peeling plane. Yellow peeling plane just because if you try to peel the bark, then you start to see that yellowish color which is underneath the bark. So that's why the name it becomes a direct translation of the color that comes out of the bark. And locally it is known as Mutabatsindi. You know, there are people who are called Mutabatsindi in the area where the species is found, and they name that species after them to say it's Mutabatsindi. And if you can look at it, it's a totemic classification. In other words, they are classifying it after themselves. And if you think of totems, people respect their totems. I mean, if my totem is an elephant, you know, in our language we call it Ndo. I know there are students here with the same surname, Ndo. You know, it means they will respect that elephant. They will never eat their totem. You know, even if we can cook an elephant here, if they are really following their culture, they will not touch that elephant. Some of us will enjoy that elephant because to us it's not a totem. So the same with these people. They respect this plant so much so that they always want to protect it. And they even go further to say, this is our species. We have been given this species by God. That is what they say. And then they even go a step further to say, even, because at times I decided to propagate, you'll see it as I talk about the research. They even go to a point to say, even if you try to propagate it elsewhere, it will never grow because it is our species. But that is a debate for another day. And of course, like I'm saying, if you can look at this distribution map, the species is just represented by a dot. This is a map from the SAAB red data list. So it's just represented by a dot. And this merely means that if this species can be wiped off, this particular population at Tengwe, if it goes, like I've indicated that I visited a population of sacred duck and then it was no longer there. If it is wiped, it means removing it from the records of South African list of biodiversity. And that, of course, we know what it means as botanists. It's going to affect the ranking of the country in terms of biodiversity. And of course, this species is already classified as a critical endangered species. So that makes it very much important, you know, to focus on. So these are just some of the uses of the species. What I can say about these uses, when you peruse through them, you can just clearly see from the onset that this species is mainly used for medicinal purposes, which are in a way further divided into medicinal aspects as well as the magical aspect. Because some of the uses that are indicated here, you can see that as a scientist, you might think it doesn't make any logic, you know, but anyway, they are still after these species for those purposes that we might think they don't make any logic. So, I mean, issues like protecting people against witchcraft, you know, protecting homesteads from evil people, you know, protection and recovery of properties. You know, when I visited the area for the first time, when I was asking for permission to work within the community, I went to the tribal authority, and then they even gave me a register, which has got visitors. You know, when you are a visitor, you will go to the tribal authority, and then they will take you to the field with their person, because indeed it's their plant. That is the person who will also dig the plant for you. And then when I was looking through the list, I realized that there was some people who came from the other province. I will not be mentioning provinces here, but those people were coming from the other province, not Limpopo. Then I said, but these people from so far, what were they doing here? And then they said, no, they don't know what they wanted the plant for, but actually their story was that they want this plant because their kumbi has been stolen, you know. And then they took the plant and then they went away. And then later on they brought back the report to say they have been able to recover their kumbi. So how they used the plants, they were not interested in that. But anyway, they used it. The last one I think is very much important, especially if you think about some of our national teams, because they are saying it can also be used to discourage opponents in sporting events. I know about this because I was once a soccer player. I know in soccer it is used so frequently, this thing of medicinal plants. They are being used so much in soccer. But as you know, sometimes you can visit a traditional healer and then you say, we need a medicine, we are going to play tomorrow. And then he can just turn back and pick a piece and then give you and say, go and play. Because you have got that piece, you will play like hell, knowing that we have got a medicine. But that thing is not right, because mentally it affects you. The day you don't see that piece of medicine, maybe you don't have the piece. Because I remember at some time when we were playing, we didn't pay our traditional healer. And then he ended up not giving us the material. And you know, by the fact that that day we were not being sprinkled with the medicine, we were so angry. We didn't play well that day because we didn't have that thing that boosts us. So it's very much important. Actually, some of the teams, they appoint somebody for special projects. You know, they will say, this person, we're appointing him for special project. And this is one of the projects that they will be being appointed for. But then the thing is, this plant has been doing well ever since, because they always claim it's their plant. But then you try to check why was this population doing well until recently, wherein they have to decide to really declare that population a nature reserve. And when you look at this, you find that there were a lot of myths and taboos which were being used in order to protect that particular species, you know. and you know, they just put some kind of social restrictions around the use of the plant, and then you start to not to use it, because they will say you are not allowed to touch it, especially active young people. They will say, don't touch it, because if you touch it, you become sterile. And I don't think there's anyone who would like to be sterile while he's still, sexually active, you know. So you will avoid it by all means. And then they will say only an old person will touch the plant. And even, especially with women, it's even worse, because they will tell you that, no, you touch it, you get into your periods. And you don't want to just get into a period willy-nilly. So what the old man in the family will do, even when he's erecting the fence around the houses, he will say, I have put also some mutabatsindi there. That is just a way of guarding the fence, because it means that even if the women of the house do not have firewood, she will not dare go to the fence, because she don't know which one is mutabatsindi along the fence. But if you don't do that, they can just go and pull a firewood from the fence and use it. So some taboos as well. These are just stories that are built around the use of the plant to say this is what happens. I mean, even this issue of saying it's their plant, I mean, it's just, when you look at it closely, you can see that it's just a story which is meant to protect it. Also, the techniques of collecting the material wherein it's only a Mutabatsindi who collected. This person who was digging here, it's an old man from the Mutabatsindi. He was digging for the traditional healer, who happened to be my friend. And then, even though he's a traditional healer, he couldn't dig it, because they will say it will not work if it is not collected by a Mutabatsindi person. So they will be putting all those restrictions to say, if you do this, it won't work. You know, and you have to perform some incantation if you want to collect it. I remember at that time when I was with the traditional healer, I wanted to collect a specimen. And then he said, no, you mustn't touch it. Let me first perform some rituals. As much as I felt I could have just collected the specimen and go, I mean, to protect his pride, I said, okay, I'll go ahead and do your things. And then he does his things, spitting some saliva, talking to that plant, and then he said, okay, now you can collect. And then I collected the plant. So the other aspect which is very much important, which is being ignored nowadays, is the issue of tribal patrol, wherein every person in the community become like a guard to that natural resource. You know, they see you in the field and then realize that you are a foreign person, then they report you immediately, and then you just see people coming to you, and then they take you to the tribal authority, and then they fine you for that purpose. So I just want to go through some of the work that I did. Some of them I did with my students, like I indicated, Dr. Tiawon was one of my former PhD student who worked on the species. So these are some of the research that we did on this particular species. In this particular one, we're looking at the population biology of the species, and then looking in terms of the stem diameter that I spoke about, we found that indeed the population looks healthy. It has got a lot of young individuals. Even the canopy was okay, looking okay, not so, because we wanted to understand if this species can be able to, you know, to survive harvesting. And then when we did the graining, we found that it's a fine grain species, which means that it can tolerate a certain level of harvesting. It was going to be worse if it was a coarse grain species, because it was not going to tolerate any form of harvesting. So this was indeed consoling to know that it's a fine grain species. And then we also looked at the size of the reserve. Here what we were trying to do was to understand whether that reserve size, which is 110 hectares, is it enough to protect this particular population? What we did, we took the map of the area, and then we grid it into 50 each and every cell to check if there are any other suitable cells around the nature reserve that can be used in case of expansion of the reserve. And then, of course, because when we look at it through the Berkman method, we realize that minimum that we need for the population to be viable is 366 hectares. And then, of course, what was interesting is that there were some cells which were found to be habitable after looking at all the risk and all the activities that people are doing around the area. And that was consoling, because then it means that they can be able to expand it if they want to do that, which is a necessary thing. But nevertheless, we also realized that at times it might not be possible to make one big reserve. Maybe they can opt for the sloth rule, which says it's either you go for a single large or several small. So these cells that I've indicated here, they can allow for that if they want to do several small reserves around the Mtabatsindi Nature Reserve, as long as they provide some corridors which will allow for the movement of propagules. Then this is what we did with my student, the PhD, former student, Dr. Tiawon. Before he commenced with his PhD, he did a review wherein he just looked at what is happening around this particular species. And then we realized that, of course, the population seems to be declining when looking at the data from 1990 and 1997. And then also on the review, it also flagged out its continuous use, you know. And not only in South Africa or in Bembe, there are a number of countries wherein it is used. But when you look at the components, the chemical compounds that are found within the plant, we realize that there are some important chemical compounds which have got some biological activities that in a way justify this continuous use of the species. And again, in that review, he also highlighted the need to understand the reproductive biology of the species going forward. And then indeed, when going forward, he also looked at the current threats at that time when he was doing his work, which was published in 2019. And amongst the current threat, of course, bark harvesting was found to be the highest threat, followed by the roots and then the cuttings. So, but when you look at this table, you can see that, of course, there are a lot of individuals that have not been harvested. But this shouldn't really tell us that we have got a good population, because this group of individuals are those individuals within the small size classes that have got no matured bark. But if you look at individuals with matured barks, all of them were ring-barked in a way. So that is something that came out of that article. And then in the other article, which was also being conducted as part of the PhD study for my former student, we look at the propagation potential. You know, again, by the way, this is the species which, while I was doing my PhD, I also tried to germinate the seeds, and nothing came out. And then with him, we subjected something like 360 seeds so that he can try to germinate them. And of course, after doing all these treatments, so that he can be able to germinate, but nothing germinated from that 360 of the seeds. And then, of course, he went for vegetative. This is wherein at least we could see some light at the end of the tunnel, because the buds did came out, although most of the budding didn't develop into leaves, but at least we saw some buds, and then we even saw some individuals with developed some leaves, which means indeed there is something here that we can rely upon. And because he was using the soil from the site in the agromix, what was interesting was that the soil from the site performed much better as compared to the agromix, which then in a way makes you think again about the comment that they've been raising to say that this is our plant, it will never grow anywhere. Maybe because we took a piece of their land to the lab, maybe that's why it grew, I don't know. But they maintained that one. Of course, the hormones that was found to be performing better was the IAA, so that was that. And then I also wrote a paper around the management plan of that particular species. And when I was looking at the management plan, after considering all the stakeholders within and around that population, I realized that it is important maybe to come up with an integrated management plan wherein you will try in a way to involve all the stakeholders, including the nature reserve management for that matter. And also acknowledging the fact that the nature reserve is found within the biosphere. I'm sure all of you by now you know that Bembe has been declared a biosphere by UNESCO. So within a biosphere reserve, you know there are zonations, your zones, your area. In a way, I see it as a flexible way of conserving natural resources, because it allows for all of these activities as long as you restrict them to certain zones. Like your transitional zone, you allow almost people to do all the things that they want to do. And then when you go to the buffer, you start to caution their activities until you, and then people feel free working around such an arrangement. And we're saying this kind of arrangement, it can also do well if you're looking at it in terms of ecological solidarity, concept of managing natural resources using this community-based natural resource management system. And of course, lastly was the fact that conservation plan should also involve, you know, when we talk about land use options, let us also think about the characteristics of the species, you know, in terms of classification. So that should also assist going forward. So after doing all these things, we realized that, of course, we still have the propagation dilemma. I will talk about this later on again. But then, going forward, then, the same tree, you know, this was then. They used to harvest the bark, you know, and leave a tree standing. And because this plant has got that kind of resistance, can grow back the bark, it used to grow back the bark and then seal the whole tree. Because they want to take all these barks. Now they cut the whole tree, and this is the trend now. People are just cutting. But they were talking about things that are happening within the reserve, you know. So they just get in and cut. At the moment, the reserve people knock off, because of course they do knock off. When they knock off, there are no securities that are guarding the reserve, then their shift begins. And then, the shift of the poachers begin, and then they go in and poach as much as they can. So now they're cutting down. Yeah, the other thing that we felt should also be looked at is the issue around pollination. I think this is wherein probably we also need collaborators who can assist us. This is some of the work that we are looking at, so we don't know at what stage it is being preyed upon. So that might need to be looked at. So I think I omitted a slide. Oh, this is the slide that I omitted. In terms of the future research around propagation, is the issue of seeds. Like I said, we are not winning, and our eyes are turning blue when it comes to seeds germination. We are not winning at all. But nevertheless, it doesn't mean that we should give up. I think the resolve should be to continue doing what we can so that we can save the population. Of course, the issue of cutting, of course, we can see now that there's something that can be done around that. And also the issue of air layering technique, layering, and then we realized that roots developed. But unfortunately, by the time we opened those balls, the roots were already dry, which means there was something probably wrong with the moisturing of that medium. And now what we have done at the moment, we have put several balls again, trying to see if indeed it will yield good results. So this is something that we want to do going forward. That is in terms of air layering. By the way, this is in terms of air layering. We introduced several balls that we're trying to. And grafting is one aspect that we think maybe we can look at it, looking at some of the members within the family and then using their seeds and to germinate the plants and then graft our Brachyregia. So in conclusion, one can say that the use of this particular species is going on unabated. It's going on. There's nothing we can do about it. It has to. People need those resources. It's a fact. That's why they are getting into the reserve. Because, by the way, this reserve, when it was kind of declared, there were some individuals of Brachyregia outside the reserve. So they wiped those off, all of them, and then they started moving into the reserve. And now they are doing as much as they can in terms of poaching. So there is also a need for heightened conservation intervention, wherein maybe if the provincial government is serious about this whole thing, maybe they should think about getting some permanent securities that can be there 24/7. And of course, we are saying there has to be more focus on developing successful propagation techniques so that indeed we can save this particular species. So That's why it's suffering from this pressure. Then maybe we can assist the thing that I normally refer to as playing God. We can assist by making sure that we restore the population. We in a way, propagate as much as we can in bulk and then try to. This is just an acknowledgement of the institutions that I've worked with over the years, and I thank you, ladies and gentlemen.
Thank you, Prof, for a very interesting talk. Indeed, we use plants for medicinal purposes. We use plants for various reasons. And also, we need to protect our biodiversity. We need to protect the plants. We need to cultivate it. We need to teach our community how to conserve these plants because we don't want some of them to reach extinction. Some, they are endangered plant species. Prof, thank you so much for sharing with us the indigenous knowledge system, the medicinal use, and also, no, Prof, you are still with me. Don't shift. I thank you so much. I will allow 3 questions. 3 questions or suggestions.
Hi, thanks, Peter. I was just wondering if you can just give us an indication, how many plants are there in the population, roughly?
No, not at the moment. Not at the moment, because mostly when we were working on, we were just looking at it in terms of density, that particular area, not in terms of individual. I know such a question is very much critical, especially if you are dealing with a population that is really under stress, like Militias Pulmani. With Militias Pulmani, what we did exactly what you asked. We just went in and count each and every individual. It was easy to count each and every individual because that one is in a worse situation because there's no reserve there. But with that one, at least we still have some plants, but like I've indicated, it's just the harvesting pattern that we're worried about.
Yes, as far as I know, there are relatively few members, certainly of our South African Ocnaceae, that grow easily from seed. So that might be tricky finding something to graft on. The other thing that's very interesting is with Ocna pulchra and other Ocnas, if a major plant gets damaged by fire or something, one will get a ring of root sprouts. And what that might indicate is maybe it's worthwhile looking at trying to propagate from the roots. So doing root cuttings rather than stem cuttings might help.
Thank you, Prof. Bokon. Last question? Okay, in the absence of any question, thank you so much, Prof. We have a small token of appreciation, and also we want to thank you for accepting our invitation. It was a very interesting talk. Thank you.
Do what? To close it. Oh yeah, it is.
Our next speaker is Prof. Dave Thompson from SAAB. He's going to share with us the work that he's been doing. Also, it's about the plant species extinction. I was talking about some of the plants that reach extinction, and he's going to share with the other project that is involved at SAAB and also is NRF. Thank you so much, Prof. You may proceed with the presentation.
Good morning, everyone. Thank you so much for giving me the opportunity to be here and share some of our work with you. I have a very long association with SAAB, so it really is a privilege for me to come back now as a non-student and perhaps share with you what we've been doing. Okay, so I need to kind of acknowledge Peter. Peter really introduced a lot of the concepts that I'm going to be talking about as well today, so it's really set a nice background for that. But what I do need to say is I'm really here as the front man for a really big kind of concerted effort on behalf of a lot of people, some of whom are in the room today. So it's really nice to be able to share this with everyone. The first group of people we just need to mention is a group from WITS, two wonderful students, Kaylee and Samister, who've been doing a lot of this work. And then Glynis, Kelsey, and Ed Witkowski, and then also Yaku LaRue and Megan Kodam from Stellenbosch, who drove some of the earlier genetic work that I'll be talking about. Do need to acknowledge Sandparks. The reason for that will become clear. And then the funders, SANB, the Botanical Trust, and the NRF. All right, and so we're at a botanical conference, and I've got a picture of a cheetah, but we're not going to be talking about cheetahs. But the cheetah is a very nice model example for some of the concepts that we need to be thinking about when we're talking about species extinction. So we need to keep in mind things like fragmentation and decreased population size, which lead to physical and reproductive isolation. We need to think about things like reduced gene transfer and issues around inbreeding and the alley effect. The important point here really is that we end up in a situation where you might have decreased genetic diversity, which decreases the adaptability and the resilience of the species in face of environmental change. You may get to a point where you have some minimal viable population size or an effective population, and then if you have some kind of population bottleneck event in response to some kind of environmental stressor, you will proceed down very likely to the point of extinction. All right, and just a point to note here, it's easier to apply some sort of corrective intervention earlier in that process. It's easier and often more effective. It's a little bit more difficult to intervene in a positive way the lower down you get in that continuum. All right, and just 2 points to unpack a little bit further. The first is the alley effect, and this is really just a mathematical correlation between the density of a population and the fitness of those individuals in the population. And so you can see that for most organisms, maximum fitness is achieved at intermediate population densities or sizes. But in species where the alley effect is particularly strong, the red line, fitness of the individuals in the population actually decreases below zero at small population sizes, which has the effect of further reducing the population size. So you kind of get trapped in a negative self-sustaining system. The other point to think about, or some kind of stressor event, it could be environmental, it could be anthropogenic, which causes the population to crash. And as you would imagine, genetic diversity is very important in resisting that collapse. But equally important, genetic diversity is really important in determining the trajectory of the recovery from that collapse. Because the population here is often, or will have a much more limited gene pool than what it had there, and will determine whether the population recovers or may continue to the point of extinction. All right, so let's take a step even further back and refresh on some of our basic biology as botanists. So as ecologists, which I have sort of evolved into, we tend to focus really on the adult plants in the population, maybe the seedlings if you're feeling generous. And we run out with our height poles and our tape measures and our quadrats, and we measure things like Peter was talking about. We measure plant heights and basal stem diameters and so on. And we put together these species distribution graphs, size class distribution graphs, which show you the relative number of juveniles in the population or small individuals compared to larger reproductive individuals. And as Peter said, what you really want to see is more small or juvenile individuals and less adults. But what we do tend to neglect is the bottom half of the cycle here, where often people don't spend enough time. And that's critical because it's at any part of this life cycle that the status of a population can be determined. And so let's unpack that further. If we look at just one part of that cycle, if we look at the sexual reproduction part, if we move from the state of a plant to a plant with fruit, a lot is happening. It's a magical stage where sexual reproduction is taking place. Through the process of gametogenesis and pollination, you're introducing genetic diversity into that system, which is absolutely critical. Okay, it's critical. It increases fitness of the population, or fitness at the population level, which imparts the adaptability of that species in face of environmental change. Sexual reproduction does have a drawback. It's fairly expensive. It is wasteful and can be resource limited. And then just to point out, there's lots of other points in this life cycle: maturation, recruitment, germination, dispersal, embryogenesis, where the system can fail as well. So there's a lot of places in the reproductive cycle where the fate of a population can be determined. And then we would be remiss rather not to mention that other form of reproduction which is available to most plants, and that's asexual reproduction, where you really just bypass the bottom part of that life cycle, and where the adult plants or the mature plants are really just giving rise to new individuals through the process of vegetative reproduction. The point there is that there's no genetic diversity introduced. The offspring are genetically identical clones to the parent. It's cheap to do, but you are losing the adaptive capacity of your population. And so then you get a situation where the reproductive function expressed by the plant is a trade-off between the benefits of sexual reproduction and the benefits of asexual reproduction. And it's important to realize that a species can be either of those or both of those, and that can change over different time scales. It can change over the course of a lifespan, it can change over generational time scales, or it can change over evolutionary time scales as well. Some concepts clear in our mind. Let's finally introduce ourselves to the pepper bark. It's in a small family of plants, the Canalaceae. Warburgia is the only genus in Africa. It's a small to large tree. It occurs in montane and sandfeld forest and wooded ridges on the eastern escarpment of eastern and southern Africa. It's got these small greeny yellow flowers, fruit similar to olives, and we will unpack a little bit further about that now. All right, so let's look at the African context of the species. It's critically endangered in Africa, and the primary reason for that is the very high levels of exploitation. Okay, it's an incredibly popular medicinal plant. Warburgia salutaris here in purple used to be distributed fairly widely up East Africa. It's now locally extinct in Malawi, Zambia, and Zimbabwe. The current location or localities are situated here in the black circle. In South Africa, where it's endangered, Eswatini, where it's critically endangered, and in southern Mozambique, where the species is vulnerable. And I mentioned it's heavily harvested for traditional health systems. It has a really wide range of pharmacological activities, and it's primarily the bark that is harvested. And then, of course, because of the situation, they are, due to the fragmentation of the species distribution, the isolation of the populations, and then so the ability of these populations to be maintaining their genetic diversity and their adaptation capacity. In South Africa, the plant is also very important in our traditional health systems. Recent decades, there's been a 50% decline in the South African populations, decrease in distribution, decrease in the number of to meet the demands of our medicinal markets. So in South Africa, it's restricted primarily to protected areas at this stage, Mpumalanga and Limpopo being the strongholds. That includes the Kruger National Park here, which contains or hosts one of the largest remaining populations. If we just have a look at this map here, where the crosses are, represents historic distribution, so there has been significant local extirpation across its former range in South Africa. And some of the remaining subpopulations have very few individuals, perhaps less than 10 individuals, which of course again brings us back to this idea of gene flow and genetic integrity and the persistence of the species. Just one point to mention is that the species does respond very positively following damage. It coppices very readily, and it also regrows its bark very readily. And this unfortunately has given people the sense that the plant is doing better, or is better off than it actually is, because bark, which allows people to sustain or to harvest in an unsustainable way. In Kruger, let's have a look at that. So the species is limited only on south and southeast facing rocky ridges, which is quite different to where it occurs in the rest of its range. There's 13 subpopulations, and these range from having a single individual to having several hundred individuals, and they are at most 10 kilometers apart from each other. A critical point here, the subpopulations are within, some of them are within 250 meters of the park boundary. At most, they are 4.5 kilometers from the park boundary. And because of that, there is significant ongoing levels of bark poaching despite being in a protected area. Just to show you, that's what it looks like in the field. There's a large individual and then several smaller individuals. All right, so with this species and with sort of rare plants in general, when you're thinking about conservation strategies, you're really thinking about three pillars that you can use to support your efforts. The first is multiply, the second is protect, and the third is minimal use of the species by providing material to ex situ markets. And that's been done in collaboration with various partners, the ARC, SAAB, Botanic Gardens, and so on. Using vegetative cuttings and then also tissue culture, and eventually those plants make it back to the communities. In terms of protection, the plants in Kruger have full-time guards that patrol the area, also sniffer dogs to track products that are being moved out of the park. And in terms of monitor, that's where I got involved and where SAAB got involved, was to have a look at the population in Kruger to try and detect the changes that might be occurring. And there we did sort of typical ecology things. We ran out and we counted stems. We measured how big the stems were. We did those size class distributions, and we recorded the levels of poaching. And so what is the status of the plant in Kruger? Well, it doesn't have the inverse J-shaped curve that we would expect or that we would like to see for a population. The population is lacking in small or young individuals. And that indicates that there was a period in the past where the plant was able to recruit, but it's currently not recruiting, or it may be persisting by what we call the storage effect. 65% of stems are debarked in the park, and most of the stems, at least half the stems in the park, or half the individuals, are multi-stemmed as a result of past disturbance or damage. And so then the motivation for what comes next is how do we then try and understand what's going on? Well, the first thing we wanted to understand is why do we have this normal size class distribution rather than the inverse J? So we wanted to try and understand why we have this lack of recruitment. And then two ad hoc observations in the field provided further motivation. The first was that the species has an incredibly low reproductive output. You hardly ever find plants with flowers. You almost never find plants with fruit. And when you do find those fruit, they are heavily parasitized by marula fly. and we have never found seedlings in the field. And then thirdly, when we were in the field, we noticed that where the roots are exposed or damaged, like in this erosion gully, the roots respond by producing root suckers. And this sort of fitted nicely with some anecdotal reports from KwaZulu-Natal where people had reported that they thought that the species might be reproducing clonally. And then so what we aimed to do was really unpack the antagonism between the sexual and the asexual function in the species, to try and understand what's controlling the sexual output and how prevalent the reliance on clonality might be. So we first looked at that, the clonality or the prevalence of clonality, looking at the genetic structure. We try to unpack the reasons behind the failed sexual recruitment, and then we try to get sort of our hands or our brains around what would the implications be for the species in Kruger National Park in terms of its persistence. So in Kruger, we've just got a pseudo map here. This is the 13 subpopulations. This is the largest one here. We sampled 258 individuals, collected leaf tissue. 227 of those were in Kruger from the 13 subpopulations. We also took samples from 31 plants housed in different stock nurseries around the country. When I talk about individuals, we were not measuring or collecting from trees that were right next to each other. We actually collected from plants that were at least 5 meters apart to make sure that we weren't getting, well, to minimize our chance of getting material what were likely clones, and also to avoid getting material from coppice material or leaves from coppice material. And so we collected those leaves and we developed 11 polymorphic microsatellite markers, and we looked at clonal reproduction, we looked at genetic structure, and we looked at genetic diversity. And so the first thing in terms of the clonality, we looked at tested for linkage disequilibrium for linked loci in the population, and evidence for prevalent clonal reproduction within Kruger. In terms of genetic distribution, if we look how those clones are distributed among the populations, there's our pseudomap again. So in Kruger, we sampled 227 stems, but we only turned up 114 genotypes, okay, which means that at least half the stems in Kruger share their genotype with at least one other stem. Okay, and those genets varied in size from 1 individual to up to 23 individuals sharing the same genotype. If we look at some examples, there was one subpopulation. All 14 stems that we sampled had the same genotype. Okay, the entire subpopulation where most of the individuals were unique. In the largest subpopulation we sampled 100 stems and turned up only 25 genotypes. Okay, so 75% of those stems share their genotype with at least one other stem. Stock populations were fairly diverse. Only 24% of stems shared their genotype with anything else that we had sampled. It gave us a range of diversity estimates, including Shannon's diversity. And paradoxically, we turned up high levels of genetic diversity. And this was kind of like a red flag in my brain. Like we didn't, or at least initially didn't understand why this would be the case. Just some examples there. There's two of the larger populations we sampled. High levels of genetic diversity. Okay, but how we've interpreted this is that there are a high number of unique genotypes within their subpopulations. They are also high within genotype accumulation of mutations because the genes are not in circulation within the populations. And it can also be that there has been the loss of sexual function is relatively recent. Just to kind of confirm that, there was our subpopulation with the single, where all the stems have a single clone, and we turned up zero diversity there. If we kind of put that all together in a single figure, we get the minimum spanning network of similarity. Just important points here is that the wild population, single individuals with single unique genotypes. The larger nodes is where genotypes were shared among multiple stems. So the turquoise dots are the stock plants, the plants taken from outside the park. An important point to take from here is that the stock plants are mostly single individuals. few cases where they weren't, but they or separate from the wild populations. Okay, the genetic structure, the diversity in the park is not captured in the stock populations. So how does this look in the field? Well, there's our largest subpopulation. We sampled 100 stems out of about 300 or 400. So that's them sort of plotted as individuals in the landscape. But if we plot that then as clones, we see that the population now consists, instead of a linear population of individuals, we now have a series of clones running down that ridge where these are color-coded, so dots or shapes with the same color represent stems with the same genotypes. So you've got a series of clones rather than a population of individuals. Okay, so moving on then to the second component. The second component was to unpack the reproductive ecology, try and get a handle on why we have this failed sexual reproduction. Populations indicated there in green, as well as one small population that was actually discovered after we did the genetic work, where old fruit were actually found on the ground under the trees. The flowering period is in June, the fruiting season in summer. So we turned up 42 trees that produced flowers. And we used the flowers on those trees to conduct a range of breeding system experiments. We tested for autonomy, geitonogamy, outcrossing, and then natural pollination, bagged and unbagged, to try and protect from the parasite, the fruit fly parasite. Autonomous selfing didn't result in any fruit initiation, but even the other breeding system experiments resulted in very low levels of fruit initiation. Okay, mostly around less than 5%, a little bit higher in some cases. So that's very low levels of fruit initiation. And then the only breeding system experiment that we conducted, or that we noticed, was that natural pollinations were the only pollinations that resulted in fruit set, or fruit maturation rather, and at incredibly low levels, okay, less than 3% fruit set. So we have a population where fertilization is low and abortion rates are really high. In terms of pollination syndrome, this was actually not documented for the species. There were some suggestions because of the structure, the color of the flowers. It could be solitary bees or wasps. We turned up a range of butterflies and moths, flies, beetles. We did get some wasps and bees, and also thrips. But based on the abundance of the pollen, the contact between the pollinator and the flower, and the pollen loads, we're really confident that the primary pollinator of the species is the moths and the butterflies. Male function, we had a look at that in terms of pollen viability and pollen tube growth. Viability is fairly high, anywhere from 60 to 90%, depending on which subpopulation we collected the pollen. Pollen tube growth, irrespective of where we collected the pollen, about a third of those actually initiated tube growth, but only about 7% of those actually reached the ovules. So pollen production is not that low. It's relatively low, but not that low that we'd be concerned. But in one population, we noticed that although the flowers produced anthers, the anthers never dehisced. And so the pollen was never released, and that is actually a documented form of genetic male sterility. In terms of seed and fruit set, okay, we only found fruit in the largest population. In 2019, 2 stems of a single clone produced fruit. In 2020, those same 2 stems produced fruit, as well as 1 stem of an additional clone. 9% of fruit that are initiated, you can't really see it there, but there's a little hole at the base of the ovary. Something is damaging the ovaries early on. The fruits are aborting. 20% of the fruit that do start to mature are parasitized by the marula fruit fly. So again, we have this situation where we have a population where there's these massive levels of abortion post-fertilization. About 91% of fruit that start initiating are aborted. And there's 2 possible reasons for that. The first is a resource limitation. The second is the activation of some developmental lethal in the form of a late-acting self-incompatibility or post-zygotic failure. But at the end of 2 years and a lot of work, we did manage to find 22 mature fruit. We extracted about 50 viable seeds from those fruit, and those were germinated and have gone into the Skukuza nursery stock plant population. So what does this mean? Okay, so we've got, what is the reproductive potential here? If we define reproductive potential as the ability to set mature fruit with viable seed, in the pepperbark in Kruger, our reproductive capacity is 22 fruit from 3 stems of just 2 clones. Okay, so the interpretation of that is that most of the genotypes in Kruger have lost their ability to reproduce sexually. And it doesn't have to be that way. This is the thing. Elsewhere in its range, okay, this is now an extralimital population established in KwaZulu-Natal. Trees were planted about 30 years ago. They are massively prolific. Hundreds, thousands of fruits produced every year. Okay, so then these fruit have been very important in the last couple of years because they are underpinning all the multiplier efforts that are happening around the country to get plants into nurseries to give back to the communities. All right, so what does this all mean for the species? So the species does have reproductive, well, as a species, it has reproductive potential, but it is fairly limited in Kruger, but we do have sunflower production. Pollinators are not a problem. Male function is not a problem, and those seeds that are produced are viable. Okay, but the problem is coming in the chronic lack of mature fruit being produced. We have these low fertilization rates, low levels of embryogenesis, and really high levels of fruit abortion. And so I mentioned before the 2 possible hypotheses there. The one is the developmental lethals. And so we've shown the species is primarily outcrossing. But because the pollen is not in circulation, because the nearest neighbor where the pollen might be coming from is actually a clone of yourself, the species has been forced to self-pollinate. But we showed that it is not capable of self-pollinating. And the second point is the resource limitation hypothesis, where fertilization may be happening, but the plant is allocating its resources away from sexual reproduction. We have probably the relatively recent failure of sexual reproduction in these populations, and that the species is almost certainly persisting via clonal reproduction in the park. And the problem there is that the longer you reproduce clonally, you further lose your ability to reproduce sexually going forward. Okay, so what is the fate of the pepperbark? We're getting to the end here. The populations are fragmented and decreasing density. There's possibly the alley effect coming into play. There is reduced gene transfer, but that diversity is not in circulation in the population. So we've established that the effective population size is very much smaller than the standing stem population size. And so the question is, have we got to a point, do we have a minimum viable population? And will that withstand some kind of population bottleneck? And so just to finish off, we're going to look at interventions here. Well, first let's just have a look at that in terms of a population bottleneck event. So in mind with the Kruger population, I don't think it would take much to initiate a bottleneck event. It could be a drought, a series of droughts, something like that, some kind of environmental stressor, which is going to get us to a point down here where it's sort of unknown or unclear whether we would be able to recover or proceed to local extinction, which would then remove one of our largest populations from the distribution map. Okay, so what are some of those interventions? Well, we're going to come back to those points we spoke about earlier: multiply, protect, and monitor. Okay, so in terms of multiply, stock plants are diverse, but the Kruger genotypes are not captured in stock populations at all, so that's a priority. You need to get those genotypes into ex situ populations. Focus on getting plants from seed rather than through vegetative means, and because we now understand a little bit about the pollination syndromes and the breeding systems, we can now do targeted searches, targeted pollinations, bag fruits, and so on to try and maximize our ability to get viable seed. In terms of protecting, We need to expand our concept of protection. We're not just protecting the plants from bark harvesting, we need to protect genetic diversity as well. That, of course, is driven by resource limitation. I'm talking about financial resource and manpower. So we probably need to think about establishing gene banks and possible rewilding programs. Monitor. We obviously need to keep on monitoring. We need to assess the Kruger population relative to other populations. We have established the effective population is much lower than what we thought in terms of stems. And we need to take the outcomes of the earlier monitoring, we need to kind of discard that, because a population persisting through clones is not going to behave in a way that we expect a population reproducing sexually would. We need to, of the life cycle, not just the diameter of the stems of the standing individuals. And of course, we need to do this over the long term, because any intervention we put into place now is going to have some kind of lag effect that we would need to be looking for. And then very lastly, we need to change our mindset. Okay, if we were reporting these levels of reproduction and these population sizes for something like a cheetah or a rhino, the species would be getting a lot more attention than it currently does. I think we do need that the Kruger population does have some defined endpoint. I do not think it will persist indefinitely. We need to think about the species. We need to be interested in the species more than just because it's a medicinal resource. Okay, there's a lot to learn here about the reproductive systems and so on of threatened species. And then what my involvement here has really driven home for me, and what the previous talk as well highlights, you need to have clear research questions to help you understand what you're observing in terms of the monitoring, and to also guide your multiplier efforts as well. An example of that is just a project that we're busy with at the moment, looking at climate niche models to try and help us unpack that resource limitation hypothesis to see if the species is in fact at disequilibrium with its environment. I do need to thank a lot of people. I'm not going to go through all of them, but the two that do need to be mentioned by name is two field rangers, Simon and Venus, who patrol those plants every day. They know the plants intimately. They were the ones who helped us find the plants with the flowers. They were the ones who helped us find the plants with the fruit. Without them, none of this would have been possible. So I do need to acknowledge them. Thank you very much.
Thank you, Dave, for a very interesting talk. I've learned a lot about the levels of reproduction and also the bark poaching. We talk about the rhino poaching, but it's also very important to understand why we experience bark poaching and so on. I'm going to allow 2 questions, but before that, Dave, I just want to say that when you were presenting, I've seen the area where some of us, we can collaborate. We have our postgraduate student who presented, some that are still going to present today. From my institution, from the Faculty of Science and Agriculture, I've already identified some of areas. And to the delegates, this is a good opportunity for us to identify those areas of collaboration. Let's just use this platform to make sure that we strengthen the collaboration for our postgraduate students and also for us as researchers. Thank you so much, Dave. I'm going to take only 2 questions.
Dave, thanks a lot for a great talk. It was really interesting. So you ended with these 2 hypotheses. You had the idea that maybe fruit set or seed set was limited by resources or by partners, I think which you called the Ali effect. And you suggested testing the resource limitation idea using niche modeling, which sounds like a great idea. I was wondering if you could test the partner limitation idea by bringing in pollen from another population, and would that be something that's desirable to do in a park, I'm not sure, but it could maybe solve your problem.
So it is something we've spoken about. It's just a case of finding the budget at this stage to support something like that. And it certainly it would be a nice add-on student project. Sorry, Glynis is just saying she can't hear. But the suggestion was, could we get to the sort of unpack the genetic hypothesis for failed sexual reproduction by importing pollen from elsewhere? And we have spoken about that. And I think it is something we should pursue.
Okay, thank you. Last question. Carmen.
Thank you. The ones that were reproducing so freely in KwaZulu-Natal, did you check the genetics of those? Are they clonal or not?
So the population in KZN, the extralimital population, they were included in some of the stock plants that we included. So their genetics are distinct from the Kruger population. But mostly they were, we don't know if they are clonal or not. That would be something we would have to test as well. The origin of the population seems to be from False Bay at St. Lucia, and seemingly came from seed that was collected about 40 or 50 years ago and taken to Silver Glen. So it was originally from seed, but how diverse those seed was, I don't know. Yeah, absolutely, yeah.
Thank you so much, Dave. We have a special small token of appreciation. We just want to say thank you for accepting our invitation. I know you had a tight schedule, but thank you so much. A round of applause for Dave. Our next speaker, next international speaker is Prof. Alan Goosen from Ghent University, Belgium. He's going to share with us about the jasmine hormone. It's a very lipid-based plant hormones. He's going to share with us about the project that they are doing at the Ghent University in Belgium. We are looking forward for your presentation, Alan. Thank you. Welcome to South Africa.
Okay, it works. Okay, first of all, thank you very much, Salome, for the nice invitation. Thank you also to all the society for a very warm welcome. For me, it's the first SAAB meeting, and I hope to have many more, maybe. Who knows? Okay, maybe a short introduction on myself. I work in Belgium at the VIB, Ghent University. It's a kind of a hybrid institute which actually combines applied and fundamental research. So my work deals with how plants make bioactive metabolites. We want to understand that so we can also exploit them better. Exploiting meaning producing the active ingredients, those plants at higher amounts. And for that, I decided a couple of years ago to focus on jasmate, a hormone. And actually, you may not have noticed, but every day when you pass by or you walked from the restaurant to this meeting room, you were actually smelling jasmate all the time. So it's all everywhere. So, what's the connection between jasmate and metabolites? I will try to, is this working properly? Okay. I will try to illustrate this with the coming introductory slides. So, as you know, and as we have seen in this meeting a lot of times, plants make thousands of different metabolites. A single plant even can make thousands, and if you combine the whole plant kingdom, you have hundreds of thousands of metabolites. These metabolites have enormous diversity in structures and bioactivities, and that is because the plant constantly has to face a changing environment, and since they cannot run away, the only thing they can do is make chemical substrates to actually defend themselves. They have to defend themselves against pathogens, to the poaching probably as well, and also to all kinds of abiotic stresses like drought, salinity, high light, and so on and so forth. So that means that metabolism is changing all the time. So plants need to orchestrate this very well, and for that they have these metabolic circuits. And this is, for instance, the metabolic circuit of tomato, very elaborate one. Thousands of metabolites again, with thousands of enzymes involved. And I want to show you how dynamic this metabolome is, and that also has consequences if you want to harvest actually plants in the field and find your active ingredient. So tomato in the green stage, it still needs to, or the fruit at the green stage still needs to proliferate. At this stage, it should not be eaten before it's ripe. So at this stage, you have, of course, chlorophyll being produced to capture the light and carry out photosynthesis. But the green tomato fruit also makes these very bitter and toxic glycoalkaloids, with alpha-tomatin being the main one. This is at this stage. When the fruit is ready and the seeds are ready, seeds need to be dispersed by herbivory, by birds, by animals. So actually at this stage, the fruit becomes more attractive to those animals, and actually there's a whole reprogramming in the circuit. This means that now you will get carotenoids, acyl sugars, phenolics, and the bitter alkaloids are converted to a more sweeter version called esculosides. So this makes the tomato fruit more interesting for seed dispersal. Now, this is not a given fact. At any stage, this circuit can be reprogrammed if you have any kind of stress taking place. So in this case, if you have herbivory by, for instance, caterpillars feeding on the leaves or the fruit itself, the plant will start making jasmate, and jasmate will actually reprogram the circuit completely. You will have an arrest in photosynthesis, and basically now you go back to these bitter alkaloids, you get reprogram of primary and central metabolism, and that leads to the extra feeding of these defense metabolites. It's not just that, also the smell and the fragrance of the fruit will change. They will produce volatiles, and these volatiles will attract now other organisms that will help the plant to fight the caterpillars. In this case, parasitic wasps that will put their eggs on the caterpillar, and then the larvae will actually literally eat and digest the parasite for the plant. Now, if you're, for instance, interested in alpha-tomatin, this is typically, although we taste it and bitter, it's not present in high amounts. So if you want to increase alpha-tomatin levels, you have to actually know the whole circuit. If you just touch engineering, you need to understand this. And like 10, 15 years ago, I decided that jasmate could be key here, and this is why I will spend also the rest of my talk on explaining you how jasmate actually is important in the reprogramming of the circuit, and how it has both interest for engineering, but also for ecological aspects. So what do you need to know about jasmate? I will try to give a kind of a short molecular introduction. So jasmate is produced by every land plant, which means that they all have this molecule, and they all have a receptor for that molecule. That's everywhere present. Now, every plant makes a totally different metabolite, and actually a mix of metabolites, and that is because downstream of this receptor complex, you have a very specific molecular machinery, and that each time has defined a very specific metabolic set for each specific plant. So if you take tobacco, nicotine, you can increase the levels of nicotine in your tobacco if you apply jasmate. The antimalarial compound artemisinin, again the same thing. Tomatin and tomato, again the same thing. I also browsed a bit literature before coming over here to see if people in South Africa were playing with jasmate. Not so much yet. So these are the only 3 species, but yet they're interesting. So cancer bush, the South African geranium, and devil's claw, they all produce higher amounts of their bioactive metabolites if you treat them with jasmate. Now, we go a bit deeper. What's below this receptor? Actually, it's all about transcriptional regulation. And I don't want to go into hardcore molecular biology here, but I do want you to appreciate these 2 important elements. So these can be switched on and off if you have your stress condition. So the off and on condition is depicted here. So basically, if your gene is off, it's actually located on the genome in a very condensed form, not accessible to the transcription factor or the machinery that will activate the transcription of your enzyme encoding gene. If there's stress, in this case, you will have opening of the chromatin. So-called transcription factors can bind to the gene encoding your enzyme. Those transcription factors will recruit this mediator complex. The mediator complex will actually recruit the RNA polymerase, and then you have transcription of your gene encoding the enzyme, your enzyme is being produced, and your metabolite is being produced. Now, in the case of jasmate, it's actually, you have to take into account that jasmate is a stress response, but if the stress is gone, your plant immediately reaches to reprogram to growth. So this is very well programmed at molecular level. So this is the situation for jasmate. If in the case of a happy plant, you have low or no jasmate produced, and then you have this hallmark transcription factor, MYC2, which is bound by, you can call it a concrete block of proteins that stick on the transcriptional activator, and that blocks recruitment of the polymerase. If you have stress, herbase for instance, you have instant production of jasmonate that leads actually to the activation of a machinery through the receptor that will degrade this concrete block, and then you will get actually release of the transcription factor. It can recruit the transcription complex, and you will have your enzymes being produced and your metabolites being produced. If the stress goes away, immediately there is a kind of feedback loop installed, so immediately you will also have increased transcription of the concrete block. So again, when the plant is happy again and it has successfully defeated the pathogen or the herbivore, you will have immediately again blocking of the transcription factor, and you go back to the situation, and your plant will no longer produce extra metabolites. So it's, when you want to understand it, it's all about understanding the molecular interactions. Proteins with metabolites, proteins with DNA, and proteins with other proteins. So the past 15 years, I have been working with all kinds of so-called omics platforms to understand this. And I'm not going to go too much into detail, but I want just to give you 2 examples of what we found. And I will work with, or I will show work that we did with bioactive triterpenes as the model. 2 stories, Ardopsis thaliana, which makes a model which makes bioactive triterpene subponents. So a bit of basics. So basically, all living organisms make this molecule, IPP. All eukaryotic species, animals, fungi, plants, make oxidosqualene. And why do all eukaryotes make this? Because actually they use this oxidosqualene as a building block for sterols. So we build cholesterol from oxidosqualene, fungi build ergosterol for oxidosqualene, and this is essential for membrane functioning. Plants make slightly different sterols, but again, essential for membrane functioning. Now, plants became much more inventive in this pathway. They actually made lots of duplications of this gene. So they have lots of similar enzymes, which actually do something different with this building block, and then you get other kinds of molecules which start playing defense roles. And in Ardopsis, this leads to a particular compound called thionin. And as you can see, the structure is quite different from the building block. And that is because there's a whole series of enzymes involved in the conversion of this building block to the end product, which has bioactivity. Now, in our research, we actually aim to find those enzyme encoding genes. And then typically what you do is you look into the genome and you look to genes that have actually potential activity that you would need to convert your building block into the end product. So in this case, you need oxidase, reductases, and acetyltransferases. So these can be found in the genome. And then we come back to jasmate here. What you can typically then see here, just focus on the left one here, the wild type, you can actually see that if you apply jasmate, this reflects the enzyme level. You will have higher amounts of the enzymes in presence of jasmate, and in this way you can actually distinguish the potential proteins involved in your pathway and from those that are not involved. If you then check in your mutants, for instance, that don't have the receptor, you will see there's no activity whatsoever in response to jasmate. The same at the metabolite level. Again, if you apply jasmate, you get much higher amounts of the compounds that you want, and in the receptor mutants, they are totally not produced. So if we then have our set of candidate genes, what we actually typically do is create mutant lines or transgenic lines where we actually have higher activity of the enzyme or no activity whatsoever anymore. So in this case, for instance, if you knock out the first step in the pathway, you will see that some peaks will start disappearing, and other peaks, in this case the intermediates, will have higher accumulation levels. If you do that systematically for the whole pathway, you can actually then recompose or reconstruct the whole enzymatic machinery involved in the biosynthesis of your bioactive metabolites. Now, in the next slide, I want to come to the ecology. What people may not know is that if you take your plant and you make an extract, you're actually mashing up all kinds of different organs, all kinds of different cells, and actually you just make an artificial mix of things that actually are never sitting there in the same cell. So if you go down to the situation for Arabidopsis roots again, you will see this is a kind of a so-called marker line where we actually tag one of the enzymes to a fluorescent protein, and then you will see that the enzyme and actually the whole pathway is only active in the two outer layers of your roots. Basically, nowhere else in the root you get production of your bioactive compounds. And actually that matches very well the metabolic profile, because if you would just dip your roots in chloroform, actually most of your triterpenes will come out, and most or less will actually stay in. Also, if you look just in your medium or in the soil, you will also detect the presence of your bioactive metabolites. And that is then clearly seen if you, again, go to your collection of mutants, you look at them, you knock out enzyme one by one, and then you look at the microbes living in the rhizosphere, and you will see that the composition of your root is totally changing. If you then purify your metabolites, and again, you test them one by one, or as a mix against all kinds of pathogens, or beneficial microbes, bacteria, fungi, whatever, you will see that some promote as a red growth, and others will actually block growth of certain microbes, and this causes then the changing in the rhizosphere, and actually showing that these compounds have very important ecological role in the survival of the species. So for the next story, I want to move on to Medicago turritella. So this species makes, again, a different molecule from oxidosqualene, in this case beta-amyrin. Beta-amyrin is then oxidized by so-called P450 enzymes, sugars attached to it, and by this case they become the so-called saponins, which you can also find, for instance, in soap. Now, this is a very simplified version. If you would look to a total root extract of this plant, for instance, you will have, of course, plenty of flavonoids. The species is known for that, but you will also have hundreds of different saponins again. You can maybe think why a plant does need so much of these saponins. Well, actually, it provides a very evolutionary advantage. If you have a big gamma of different molecules with different structures, you will actually have increased resistance to evolutionary adaptation of potential pathogens or herbivores. Now, I want to go a bit more in detail to this pathway. So there's a very important aspect of this pathway. It actually, after this first molecule, it branches into branches. The non-hemolytic branch and the hemolytic branch. The hemolytic branch is actually the most important in terms of defense, and it's actually it's called hemolytic because of this dual nature of a hydrophobic backbone with all the sugars attached. It can actually permeabilize membranes, and this is not the case for the upper part of the pathway. So these are the most bioactive ones, and they're actually very toxic basically to any membrane of any eukaryote, including the plant themselves. Now, of all the compounds that is produced by Medicago, this one is actually the most toxic or the most bioactive one. Yet, you will hardly find it in the plant. The plant stores it in a less active form. In this case, with much more sugars attached to the C28 position, which is very typical for these molecules. So basically, the plant has a fantastic defense metabolite, yet it does not produce it in high amounts, and it actually deactivates it. And in the next slide, I will explain you why it does that and why it's important. So back to jasmate again. So this is, for instance, cell culture from Medicago, but you can also do the same with plants. If you apply jasmate, you will have within 24 hours an enormous increase both in the number and in the amounts of your tritopene components. This is again mediated at the transcriptional level, so yellow means no expression, blue means high expression of your enzyme encoding genes. You will see that within 4 hours, all of your pathway genes encoding the enzymes are again activated, and that leads to the increased amount of your molecules of interest. Again, this involves this machinery, and what you also see is this amplification loop here. So basically, you will also find the transcription factors involved, and these are activated even earlier. Now, in the case of medicago, we noticed that there were 2 extra transcription factors next to the central one that I mentioned in the beginning that seem to be very highly co-regulated with all of these pathway enzymes. So what we then typically do, because this could be a potentially interesting tool for metabolic engineering, so if this is really a master activator of your pathway, you can overactivate it, and then your plant will make more of your bioactive metabolites. So what we then typically do is work with so-called hairy root cultures. I think it was mentioned once already here on the conference. And here we exploit a pathogenic bacterium, Agrobacterium rhizogenes, which has an RI plasmid, which upon infection is actually inserted in the plant genome, and this creates a very strong proliferation of roots. And I will show you what this means for medicago. So actually we can grow seeds, grow them in vitro cultures. We cut away the primary root, we infect with this agrobacterium, and we add extra plasmids here with the genes of interest that we have. And then you will have new roots appearing, and the pink one here illustrates this one. So this is again, for instance, one with the fluorescent transgene. So you will actually get now transformed hairy roots. And with medicago, you can actually cut off these hairy roots and put them in bioreactors, and then you can create tons of your plant root of interest, and then you have a very sustainable production platform for the bioactive metabolites from those species. So what happens if we do this with these 2 transcription factors? We have the TSAR1 overexpression line, and we have the TSAR2 overexpression line. If you go back to the branching of the pathway, the hemolytic and the non-hemolytic, you will see that TSAR1 activates nicely the enzymes involved in this branch, and TSAR2 nicely activates the enzymes in this branch. Same, this is translated at the metabolite level. The TSAR1 is activating only increasing the accumulation of compounds from this branch, and TSAR2 is activating the production of the compounds in this branch. Now, back to the, let's say, less and more toxic version of the metabolite, and how that links with this research. So we noticed, as I mentioned, that we find this molecule, which is less active, but we're actually more interested in this one. What we also noticed when we were looking at the genes regulated by jasmate and this transcription factor, TSAR2, was that there was a so-called beta-glucosidase called G1 that was actually always co-expressed with the enzymes. But if you look to the pathway, there is no need for such activity. What this gene or what this enzyme does is actually reverting this reaction. So the reaction from the more toxic to the less toxic form is catalyzed by a glucosyl transferase. The inverse reaction would be catalyzed by a glucosidase. So we cloned the glucosidase, we made a recombinant enzyme of that, and now we applied that to metabolite extracts of medicago roots. And what you see here is the black peaks are things that disappear when you, the black peaks are proteins, metabolites that disappear when you treat with your bioactive recombinant enzyme. The yellow peaks are things that appear. And to make it more simple, what we see is that when we apply the enzyme to a root extract, we actually see reconversion of all the metabolites that have this extra glucosylation at this position to the more bioactive version of your metabolite. So then we had a very unexpected finding. So if you would think that the plant needs to make this bioactive version of your metabolite, so it can actually make the bioactive version. Now what we found, and this may be a bit unclear for you, so the yellow dots is again the glucosidase attached to a fluorescent tag, and this yellow dots is actually this little device within your cell. So it's the nucleolus, and you can say, okay, nice location for the enzyme, but actually it's totally irrelevant if you look at the pathway and the system, because the substrate is sitting here in the vacuole, separated by tons of membranes. So basically, your enzyme and your metabolite will never get into contact with each other. So what we then thought of in terms of ecology is that these molecules, or these metabolites, are actually relevant for defense against pathogens, herbivores, and necrotrophic fungi. And what you have when those organisms attack a plant, you get actually cell damage. And in this case, we try to mimic that by very, I would say, naive. We just mash the roots and we look what happens with the location of a protein. It can disappear. But what actually happened was that it was relocating all over the cell. It's a bit faint here, but I hope you can see it. And then we also looked at the metabolite level, and we compared, again, mutants and overexpressing lines. So this is the substrate of the glucosidase. This is the product. What you can see here is if you actually treat or just damage the roots, you will get consumption of the substrate, and you will get increased amounts of your product, in this case, the more bioactive version. If you work with a mutant which no longer has the enzyme, you will actually have no conversion whatsoever. You even have more higher amounts of your substrate, and you get nothing of the bioactive version. If you have a more active line with more of the enzyme, you get actually much higher production of your bioactive metabolite. Going back to the ecology, when we now look at how these mutant lines behave in presence of necrotrophic fungi, for instance, Rhizoctonia, then you can nicely see here that if you, this would be the level of infection of a chrontorline, so which has the bioactive version of the enzyme and the bioactive version of the metabolite, you get a basic level of resistance. If you now have a mutant line which no longer has the enzyme, it actually becomes totally susceptible to your necrotrophic pathogen because it can no longer make the only metabolite that matters to fight actually the fungal pathogen. So this brings me to the final slide and the model. So here you see a wild type situation with the enzyme sitting in the nucleus and your product in the vacuole. When you have necrotrophic pathogens, fungi for instance, attacking, you will have a release from the product from the vacuole and release from the enzyme from the nucleus. They will get into contact with each other. You get the bioactive version of the metabolite, and those will actually literally kill the fungus by destroying the membrane of these fungus. If you have your mutant line with no enzyme, the fungus can do whatever it wants, and it will not be affected whatsoever by the present machinery that looks very promising but actually has hardly any effect. So this is the last slide. Nox, I hope you apologize me for stealing some slides from your presentation yesterday. I thought this story was relevant because people always go looking to the field for bioactive metabolites. And of course, there's resource limits. You cannot go every day at different times of the day across different seasons. But basically, if you would harvest a plant for one day from one site and you would go back the next year, maybe you have totally lost the interesting activity that you found and you have to start over again. Basically also, even treatments can make a difference. Your plant may be infected by that pathogens, and that may actually cause certainly your plant to be much more interesting in the, let's say, the bioactivity assay that you were performing. So with this presentation, I hope I have convinced you that jasmines could also help here in this screening of South African medicinal plants and to do the funkiest biotech research that Nox was yesterday referring to. And I hope to see some more jasmine papers from South African plants in the future as well, because it was very limited to my surprise. So this is my last slide. I want to thank the people involved, especially my group. Many of them that were involved in research shown have left. Some of them are still there. Elia is the one who did work on the glucosidase, for instance. And then I have collaborators all over the world. In this case, Anne Osborne for the Aropsis work, Monica Höfte for the fungus work with Medicago, and Tessa Moses for all the profiling of the triterpenes that we look at. And of course, the facilities at the facility and the sponsors. And with this, I thank you for your attention.
Thank you so much for a very interesting talk. I'm going to allow 2 questions. The last slide, copyright, Prof. Makunga. just copyright. You own the originality of the last slide. Yeah, thank you so much for sharing us with a good presentation. The tomato stories, the jasmine. We are looking forward for when we work with the South African medicinal plants. I'm going to take 2 questions. Prof. Ilof noted another one, hand. Prof. Hussein, if there's a third one, I can compromise with the third question. Prof, you may start.
Thank you very much. I think this was fascinating. I was wondering, what about endophytes? Are they resistant to jasmonate, or don't they harm the plant sufficiently to activate jasmonate?
We have 2 options. Some of them actually even activate jasmonate more to protect their plant from other pathogens. So we also found that. I didn't want to include that here because of time, but we have a recent case where we actually see that endophytes and also beneficial microbes in the rhizosphere actually stimulate jasmine signaling, so the plant makes more of the metabolites to protect itself, and actually protects the niche where the endophyte is living. You also have the opposite, of course. You have endophytes which are very destructive, but you also have, for instance, endophytes which will use the plant metabolites to convert them even in more bioactive versions that would also protect themselves. So everything is ecologically possible.
Yeah. Prof. Yeah, okay, thank you very much. You touched my heart because you're speaking about terpenoids. So you have in 3 positions the beta hydroxyl group. So we have some triterpenes which have alpha hydroxyl group. It come across to your research that's which enzyme can transform or getting alpha hydroxyl group. Thank you very much.
No, we didn't find those enzymes actually, to be honest. We looked for them because we looked at some of these funny triterpenes, but so far we were not successful. Probably we have touched the easy ones with enzymes with known functions, so you can look to the homologous versions in the different genomes. But I think we have to be a bit more creative to find enzymes that you are referring to. So no, I don't have those yet. Maybe in the future we will. But it requires a different, additional type of resources, because you have to, you're talking about, let's say, a very exotic medicinal plant species with no genomes sequenced. So basically, it requires a lot of preparative research, because you do need, and that was also mentioned by Knox yesterday, you do need this full vision of the genome to be able to recreate the network and see what the potential machinery in the plant is. And if you stay easy, like in the pathways I showed, then it's you look for oxidase, you look for that. But in your case, I would have no idea what type of enzyme, so that would be complicated.
Okay, thank you so much. A round of applause, and we have a small token of appreciation. Our next presenter, get ready. Thank you. Our next presenter is Prof. Ahmed Hussein from Cape Peninsula University of Technology. Prof, welcome. We are looking forward for your presentation. Please take us through and hear your story today. Thank you so much.
Yeah, I would like to thank you very much for the invitations. This is the third time in SAABs, the first one in Kimberley. That's where all in 2003, I think. And then in Cape Town, 2017, and that's the third one. So it's a privilege to be between you today. I'm going back a little bit historical events in drug discovery system. So yesterday, actually, we have enriched talk about the biodiversity of South Africa, and it's a little bit paining when it comes to natural products, because we are not in the level of the biodiversity of South Africa, unfortunately. So that is a sad talk for me to go through that one, because we didn't do too much in terms of drug discovery up till now. So I'm moving initially and identify my research. As you see here, we are natural product chemistry, which we are thinking about isolation identifications, and somehow we are in collaborating with other teams, botanists, biochemists, biology, whatever. And also we did some nanotechnology. If we have time, I will go a little bit some slide for green nanotechnology. It's use a natural product to improve the bioactivity of the natural product. So initially, I'm going to, that is a typical maps for the biodiversity world. This is a planet, and you see what we are living in, and you see that there are 6 flora kingdom, and that is, you know that. And this is a flora capensis, which is it has its own characteristic and its own flora kingdom. So the issue here, that green zones, this is a mega diverse countries. It's about 17 countries, and they have more than 70% of the green cover. And unfortunately, the majority of these countries are developing countries. So that is under limits, and they have no strong access or strong plan to discover their biodiversity. This is very paining, and still up till now is problematic. But we are in another phase. You are speak about 450,000 species, and the drug discovery system moved from higher plan to another system, as we will see. And this is lift that one. So for sometimes in 1980, in international level, there are a lot of regulation and conventions to start to study the genetic sources in the world. And there are one born, what's called ICBG group, is an International Cooperative Biodiversity Group. This is supported by NECI and NIH in the United States. Actually, this is established in the United States. And they have actually started to negotiate with a mega diverse country to put their hand on the genetic resources. This is another story. But anyway, they succeeded actually to start the project in many countries. And some of them succeeded, some of them they stopped at some phases. So they started, for example, in Suriname and Madagascar here, in Panama, phase 1, 2, 3, and there are some Brazil, Argentina, they stopped, they didn't continue, and somehow in South Africa as well. You don't know about this one, but we have ICBG project as well here in South Africa. And it's a well-designed group, very restrict, and they focus in discovery in bioactive molecules in at the beginning it's terrestrial plant, and they move to another resources. So ICBG, I'm taking you for one of the most important scheme of the work. This is the general scheme of drug discovery phase, and you have optimization, and then clinical 1, 2, 3, and then you negotiate with FDA after that to get the drug in the market. So, but you see this, which you don't see, this small word. So this is patentable compound. So if you have a patentable compound, it has a specific character, you have succeeded in drug discovery. If you don't have that one, unfortunately, you don't have drug discovery. So they are focused in the chemistry. So they are taking that very seriously. They have very strict plan to get that one. And I will take you for a real experience from myself, because I was part of ICB in Panama. That is, it was in 2004. And we started, it was a massive collection. Panama, it has something very important you might know, you don't know. Panama, it has a 50% covering, green cover, which doesn't exist in other place in the world. South Africa here, it has green cover, but not 50%. So the desert actually is too much here as well, but is mega diverse. So Panama as well, the very small country is about 3,000 square meter, square, and they have 50% of endemism as well. And it's green. It's fantastic, actually. It looks Amazon forest as well. So this is the steps they are follow. As I mentioned, that this is very strict. You cannot go outside. I had a lot of fight with them because I'm thinking free, but they don't so. You have to follow up the protocol exactly. So start with a random collection, test at 10 grams and 100 microgram per ml, and then go to the second phase, recollection again, as Alan mentioned phase, and you test at lower concentration at 10 microgram per ml is a kind of sieving, and then you go for priorities. You make priority B1 and 2, and then you select the best one according to literatures and the situation and so on. There are many criteria for selections, and then go recollection again in a massive, and then doing bioassay guided isolations, and then at the end you get a lead compound. That is what they are looking for, lead compound. So initially, we started with 2,000 plant materials. We ended with 10 plants. That is 0.5%. Only plant extract. We are not speaking about isolated compound yet. So you can imagine how it's frustrated and how it's problematic. So it's a big problem to work in this way. So this is my work during that 2 years in that place, and we have isolated some 34. This plant actually the top active anti-cancer plants. So we have isolated 34, 27 are very active, and 13 of them are new, because they are usually looking for new compound. And this is a typical example. One plant material is called Adinaria floribunda, which it gives resist activity at 10 microgram per ml. And then we have two very potent compounds, adrenafluorine C and adrenafluorine A. That is a typical lead compound. And I don't know what's happening after that, because that is usually going to NCI and follow up the work after that. So I will touch again in South Africa, ICBG South Africa. That is, it happens between 98 and 2006, and Vinesh Maharaj, he was at that time at CSIR, and he was leading the project, and he collected 700 plants, and they are generating 7,500 extract, and tested all of them in 3 cell lines as anti-cancer agent, and then they, after screening, they get 50 active extract. This 50 active extract, they did some chemistry, and they isolated 5 non-compound. That is banning. So after 6 years, we have 5 non-compound is useless. So that is why now is a problem, actually. It's a real problem. For ICBG, so David Coleman came in 2010 and published these articles. He said that we are playing at the end of the story. So the people which is working with terrestrial plant, higher plant, for 450,000 species, they are playing. So they said that's the end of the story. Stop working in higher plant and go to microbes and see marine natural product. So David is a leader actually in marine natural product in South Africa. He follow the international typical protocols, because at this time, 2010, United States NCI and NIH, it's changing the protocol completely, and they left the terrestrial plant completely. They dropped in the backside, and they start to go to microbial and marine resources for drug discovery. Amazing sources. I have touched few, but it's amazing, more than the higher plants. So, but they have the right, actually, to go that side. So, but it dropped 450,000 species. That's the problem. So that's the scheme again, and then we trapped in that one. As people who don't have national strategy for drug discovery, we trapped for NCI protocol for drug discovery. But I see that if we change the protocol a little bit, we can find something. So I feel that if we go to IKS system and chemotaxonomy, I think we can get something more important than the result which they got. Okay, I'm going back to the flora capensis, and I will mention historical important background in chemistry, natural product chemistry, and the people which they did that since 50 years, 60 years back, and we have a lot of people actually contribute to South African flora, and they did massive chemistry for this. So for the national level, according to Mike Davis Coleman, he mentioned that Frank Warren, he's the father of natural product chemistry. So, but I didn't see too much publication for him, because that is before 1960, 70, and there is no facilities actually at that time to publish a lot. And then those two guys, I can see Davis Rocks and Douglas Rivet, they have a few publications, especially in Lamiesi. And then the most contributor at the national level, Fanny Van Heerden, is an amazing lady, and she's doing a lot of natural product chemistry. And she's specific in saponins. Saponins, if you know natural product chemistry, is very complicated structures and needs a lot of attention. And also Daniel Ferreira. Daniel Ferreira, he was at Free State, and he moved to Mississippi now, and he made a huge contribution before leaving. But Daniel, he was only in tannins. He was affected by Mabry, Tom Mabry at that time in 70 and 80, and he was actually a good contributor, and he isolated from, for example, from Rewebos itself, you will surprise that he isolated more 70 compound, and from Mubani tree, he has a series, about 13, 14 publications. He isolated more than 100 compounds from the same plant. It's fantastic, actually. Recently he did some in Lamiesi as well, you notice, for psychoactive compound, but he didn't go to much result, but he contributed the chemistry as well. For international level, Mike, as I mentioned, he is established marine natural product. There is a lot of continuity. There is not too much student working in marine natural product up till now. The continuity is very, I don't know, it's rare. But Denzel at UWC, he take after that, but there is not too much work in marine natural product in South Africa. So for international level, there are big names. And the best and the major contributor, actually, Ferdinand Bollmann from Germany, he contributing especially for Asteraceae and intensive study for Helichrysum species, 30 species. He finished them with more than 600 compounds from 30 species from Helichrysum. And also he touched other Asteraceae. I think he reached 1,000 compounds or more, Lars Bollmann. So, Ferdinand, sorry. So the other two people which contributed to the chemistry, Benjamin Rodriguez, actually he's my supervisor. I got the PhD with him, and he had studied a lot of Belectrances species, and also Rudy from Zurich, he did also the same in Belectrances species, isolated many compounds. That is the most big names. Patricia from Portugal, he's my colleagues, and is also follow up the same route for Benjamin Rodriguez, and she's working in Belectrances still up till now. So when I'm working with these issues, I found myself that I collected a story for a set of compound from South African flora, and this set of compound, I have about 1,000 compound. Everything, the details, sources, everything about that, structures. I said, it's okay, can we go a little bit? Is doing machine learning. So I start to speak to him, and he said, it's amazing. He said, it's okay, let us start. But we didn't start actually effectively, because we just started 2 weeks back. I have collected everything as a data set, but it's very immature up till now. So we have this data set, and we started to do something similar to Chinese researchers, which they follow up a very unique study for their flora. So I said, it's okay, let us follow up their roots. And we started to do some, this is not machine learning yet, but we are start to see drug likeness of the 600 compound. And we see that the physical character of South African compound is within the drug-like phase, and it's okay. I mean that this is the features which we are selecting, but this is feature actually is a well-known and common for any drugs. So that is, if you go here for drug hydrogen bonding, donating hydrogen bonding acceptors, and this is surface, polar surface. I think it's, I can see that one. Area, polar surface area, and before there are molecular weights, and there are algorithms for solubility, and then I don't see this one. Rotatable bond. That is a physical character for any drugs. We have to know that to be within the limits of drug features. And if you go further, we have some plot scattering, and it's showing us for 3 families, which contains a major compound from South Africa, is well studied, Lamiaceae, Amaryllidaceae, and Asteraceae. So we see that Lamiaceae actually it has a unique situation among others. This is, I mean that it could be a success, a story of success. One of these is Artemisia afra. So Artemisia afra, maybe all of us touch that. And the people working in medicinal plant, doing biology, they collect Artemisia afra. Artemisia afra, it has proven to be active against many things, malaria, cytotoxicity, and many things actually. And unfortunately, they attribute the activity to flavonoid, but that's not right. You know, the key of Artemisia annua is cis-terpene lactone with endoperoxide. So you need endoperoxide, that's 2 oxygen here in artemisinin. That is the key factor in activity of the cis-terpenes. So, and there are many other intermediates, a lot, containing peroxides or endoperoxides. If you go back a little bit in literature, you find yourself, we have similar compound in Artemisia afra. We have, but we didn't isolate. After this Bollman, which he isolated this compound, after him, we didn't. No one could touch that. Myself, I worked with this one, and I tried to find the endoperoxide and even peroxide. I could not find. Following up carbon 13 NMR, but I could not find that one. It would be complexity of the structure, but it's very rich in cis-terpenes. If you see the TLC, it's amazing. But this one, it's actually is a national project. It can be taken further. And we can rediscover, getting this compound again and doing a lot of work about that one. We exceed Artemisia annua. We can go through this one. The other one is Blectransus species. It has amazing abiatin diterpenes. And this is a gland of the, if you see the leaves of the majority of Blectransus, it has gland. Some of them, they are orange in color. If you touch it, you see orange color in your hand. So this is Ecoloni, Blectransus ecoloni, and you can see the gland, amazing. So under microscope, it looks very interesting. So Barbifloron D it has a unique cytotoxic activity. And Patricia, which I mentioned her name now, he has a lot of publication in this compound. So the problem that for this compound, the bioavailability is still to be improved. But this also can be a national project, can be taken. If we are in China now, they will take it as a drug. They will solve the problem. I think that is need to be actually somehow taken into consideration. Another one is from Blectransus madagascarensis, and also this compound, it has a story with us. And we started working in 2019 on this plant to look for abiatan diterpenes, and we found that we isolated this compound, but previously it was published as this structure. And we have a problem with the stereochemistry usually. This group here, I think 1985, they published this compound for the first time from the plant, and they have 6 beta, 7 beta. This is stereochemistry, but from when I went through the publications, I didn't convince too much. So, because from the biosynthesis point of view, there is nothing. This system is not isolated before from Lamiesi. So I put question mark, and we have to go with the student. I told her we need to isolate the compound, and we isolated the compound, and we did X-ray after getting everything, and it's actually confirmed to be 6 beta, 7 alpha, not 6 beta, 7 beta. So that is actually the chemistry we have been done on this compound. But this compound, it has also a wide antibacterial activity and TB as well. It's very interesting. If you record 7-methyljaglone, that is Profilov, he will record that one. Marion Meyer, he's the first one, appoint that from Euclea natalensis roots and used for TB. The most active compound isolated from South African flora against TB is 7-methyljaglone, up till now. So interestingly, this compound at the same level, but not toxic. The problem of 7-methyljaglone is very toxic. You cannot approach that one. But this one, you can. Interesting. And it exists in Blectransus, and we have many of this one. So this is the last one. I think our colleagues from Stellenbosch, they know the story. This species is from South Africa, and they are hybrid, actually. I cannot read the names. Is Crocosmia, that is Crocosmia species, and this is hybrid between 2 species, and they are South African species. And a colleague from Canada, he came a few weeks back and gave us a talk about discovery of this compound. So in Canada, they have made a screening for 30,000 samples from NCI, and they found the extract of this plant is the most potent extract. And they follow and they isolated that Montbretin A as amazing active anti-diabetic compound. So this compound, it's block alpha amylase. I don't know if you have idea about alpha amylase and alpha glucosidase. Alpha amylase is very resistive in our assays. Alpha glucosidase is very sensitive. A lot of compound can stop that one, inhibit that one. But alpha amylase, no. We are suffering to find compound to stop alpha amylase. But this compound is a very potent, more than acarbose. Acarbose is a positive control. It's exceeding that limit. And actually, it's a story itself. The structure, chemical structure of this one is a big story, because we know, if anyone touch natural product again, we know this is glycosides, and the glycoside can be easily hydrolyzed in the stomach, because the stomach, it reached to pH 1. So it's easy to be hydrolyzed. But actually the colleagues from Canada, he said it's not hydrolyzed. It's against nature, I don't know. It's not hydrolyzed, and they detect the compound in the remains of the animals. It's amazing. So we don't know. But they did many derivatives of this compound, and also some of them, which is less glycosylated is active as well. So this species from Iridaceae, and Iridaceae is, South Africa is very rich in Iridaceae. That is open again the window for us to look carefully for Iridaceae, the corm, and look for inhibitors like this one. So I'm done with the historical background, including some of our work. In natural product chemistry in general, we have a problem, and this problem is the bioavailability. Bioavailability, low solubility, and efficiency is the most problematic issues in natural product. If you see mangiferin, for example, curcumins, quercetin, they have a lot intense work in, especially in biology, but it's not a product to put in the pharmacy, and for this compound is very difficult. This is another story, but you see the contribution. So we went for green nanotechnology. We said we can improve the bioavailability, efficiency, and solubility if we conjugate this compound through metal nanoparticles, and we selected to use gold nanoparticles to conjugate with our natural compound. So I want to tell you, we introduced this protocol for green nanotechnology long time ago, and that is specific in 96 will plate, which is it's reduced the cost. So we used a pure compound. So if you come to what's the terminology like thermodynamics and the kinetics, our colleagues in pharmacy, they are using pharmacodynamics and pharmacokinetics. It's something need to be considered, but for any drugs in health field, you need to get a well-defined drug. If you don't have well-defined drug, there is no use. That's the problem. So pharmacodynamic and pharmacokinetic to follow up the compound in the body. That is. So our colleagues, which they are using total extract for nanotechnology, they appreciate it. I'm appreciating that one, but that is not the right way to go for nanotechnology. So we deviated from that one, and we are using pure compound. We are not using total extract. So we tested total extract at the beginning, and we isolating after that the pure compound. And this is some example which we did. There are another battery in the lab. They are doing lab. The guys, hypoxicide, brucyanidines B, aspalatines, and we have 2 chalcones as well from Helichrysum foetidum, and we have aspalatine. So I will go fast. I will not go. I will not stress about this one too much. But one of the most big problems in nanotechnology, we don't understand the interface between the metal nanoparticles and the capping agent or the compound around the nanoparticle itself, the metal nanoparticle, and we start to propose some figures how it does work, and we conclude that this figure here, we conclude that under the acidic media, the aromatic system can be protonated and it generates a positive charge, can be delocalized over the aromatic system and and saturations, and these can be attracted to the negative charge of the surface, and we can fix it and making a combined stable form. That's what we thinking that it should be. And then the basic concept, we saw that the combination between the capping agent with the active compound and the metal nanoparticle actually is to change the intact active compound. The activity is quite interesting. This is one example where you see that the gold, that's the silver with hypoxicide, is active, but I think there are is potent, is close to the positive control somewhere. I can see. We didn't do positive control, but it's quite active in this sense. So I don't like too much silver for antibacterial, but it's okay. That's procyanidins B, and we get that from Leucocidia, and it's quite active as well in many assays. I won't just move. And this is chalcones. We have the nanoparticle from chalcones, which you see the intact extract is inactive, but when you make a combination with a nanoparticle, it's induced activity. That's what's required actually from that. Aspergillus linearis, it looks like a sickness for all South African, because anyone in the lab working in medicinal plant, he should start with Aspergillus linearis. So all of us touched that one. We did a chemistry, and we isolated new compound from Aspergillus linearis, and it is published, but we used aspartame for nanoparticle as well, and we did silver and gold nanoparticle for aspartame, and we did the intake to see how much the aspartame improving the intake of the nanoparticle inside the cells, and it's improving. That is my recommendation and the end, and thank you very much.
Thank you so much, Ahmed, for a very interesting talk. I'm just going to allow only 2 questions. Yeah, you're presenting a lot, plant, natural products, molecules. So any 2 questions or suggestions? Oh, okay. It looks like everyone enjoyed your talk. They don't have any question, no comment. Thank you so much for a very interesting talk. And we are just happy that you have accepted our invitation. Yeah. We'll give you a small token of appreciation. Yeah, thank you very much. Thank you so much. I just want to give a few announcements. Remember, we are live on the University of Limpopo YouTube channel. Those of you who tweet, just you can tweet at UL Varsity. Those of you who are on Facebook, just type University of Limpopo. You can get all the pictures, all the activities. Since, is it on Monday? Everything is there. It's very interesting. Just go through it and check. Even your parallel sessions, there are some pictures there. And then on Facebook, please just type University of Limpopo. Hashtag UL Events 2023, hashtag SAAB 2023. Please just go there and see the beauty of SAAB conference. And marketing team, you are doing a great job. So we are going to start with the parallel sessions. I'll give you 10 minutes to move from here. You can grab your tea and check your venue, and we start. We can start at 11. Is it fine, Prof? Yeah, at 11. Just grab your tea. You can go and drink while you are listening to the presenters. Thank you so much for attending this interesting session. Thank you.